So today's topic is nutrient media. Here you see every microorganism, every cell, they require an environment for their growth. That environment may main ingredient their nutrients. These nutrients actually consumed as source of energy. They require carbohydrate, they require protein, they require lipid, they require vitamins. All other nutrients are need to be supplied through the various sources. So if you take exactly plant cell culture, you have to remember two important scientists, that is T. Murashige and F. Skoog. Why they are discussed in detail? Because they have developed the first medium for plant tissue culture, that is MS medium. You can see here plantlets are grown inside the test tube, and did you see this jelly type substance below the plantlet? This is actually medium. This medium contain all the ingredients, and that medium and its ingredients decides the growth of plants. Is it clear? Plants obtain nutrients. Majority of their nutrients are obtained from soil. Okay, and they require carbon dioxide, they require oxygen, they require sunlight. These are the major requirements of plants. So when we prepare a medium, we have to think about all these things. The first ingredient of our medium is around 13 types of nutrients are actually obtained through soil. These 13 types of nutrients can be classified into two major groups. They are macronutrients and micronutrients. Macronutrients, they are required in large quantity. Example, nitrogen, phosphorus, carbon, etc. Whereas micronutrients, they require in less amount, comparatively in lower amount or lower quantity. We can say, for example, magnesium, cobalt, sodium, copper, they all require only in minor quantity. So first ingredient a macronutrient and micronutrients. We supply these micronutrients and macronutrients as their salts. Some salts are being added into the medium. While making the medium, we take some salts, and those salts are mixed in water. So water is a major ingredient. To the water we add macronutrients, micronutrients based on their requirement. If you know nitrogen requires in higher amount, you need to add higher amount of nitrogenous salts. We need to add less amount of magnesium salts. So these are the two important groups of nutrients which are added to this. Now another major source is carbon source. As usual, carbon source is added. This is energy source. We use sucrose, that is tea sugar, as carbon source. Then they require vitamins. The vitamins, for example, let us take nicotinic acid, thiamine, pyridoxine, myoinositol. These are vitamins. Next major ingredient is amino acids. As we all know, amino acids are of two kinds. Some are essential and some others are non-essential. A non-essential can be synthesized in the living organism's body, whereas essential amino acids need to be supplied through the medium. For example, arginine. Next ingredient is growth regulators. For example, take some example, auxin, cytokinin, gibberellin. This has already introduced you in your lower classes as phytohormones. They are actually hormones. They regulate the growth in them. Little in detail here. Toxin actually help in root generation or root formation. Cytokinin, they help in shoot generation. When you see the basic steps of plant tissue culture, growing the cells in the growth chamber, we'll introduce the cells onto different mediums, subsequent mediums for root formation and root shoot formation. In those medium, we'll be adding auxin and cytokinin. Suppose if I want to generate root, I'll add auxin in the MS medium and I'll grow the cells there. Then slowly the cells will start generating roots. These roots are again taken and kept on the cytokinin medium. Then what happens? They will start forming shoots from that. So root and shoot formation is actually controlled by growth regulators, auxin and cytokinin. One of the naturally obtaining auxin is indole acetic acid, IAA. And we can say benzyl aminopurine, BAP, is a cytokinin. If we go back, we added micronutrients. Macronutrients, all are added in the form of salts to a water, to which we added sucrose, vitamins, amino acids, growth regulators as per requirement. This particular medium is somewhat incomplete. Why? When we grow cells over this medium, we found that the growth is not proper. So other ingredients are added. These are like casein hydrolyse or coconut milk, malt extract, yeast extract, and tomato juice. This also proves that the media is semi-synthetic. That means after adding all the ingredients, the growth is not proper. So we add some other ingredients such as casein hydrolyse, coconut milk, malt extract, yeast extract. Based upon the type of medium you make, you may add any one or two ingredients from this. They supplement the growth of plants. So we can say every medium is semi-synthetic. After adding all the ingredients, we mix it very well. We may decide whether we need liquid media or solid media. If it is a solid media, we need to add some substance to make them gel. A very common substance which is used in the medium to make them semi-solid is agar. This agar is obtained from a red algae, the Gelidium amansi. Several types of medium are there, and these medium are named based on their inventors. Some other time the inventors have given certain name to that medium. MS medium, which is the most commonly used medium. Apart from that, we can have LS medium, B5 medium. The first medium is developed by Murashige and Skoog in the year 1962, and this is the most commonly or most preferred medium for plant tissue culture. After making the medium, it is very well known that it should be sterilized. Majority of the medium can be sterilized by autoclaving, but some medium cannot be autoclave. So for that we can go for ultrafiltration or chemical sterilization. This is not done for all the medium. If MS medium can be easily autoclave. But if we add some ingredients to MS medium other than that, those ingredients are heat labile. Mean heat labile means they may destroy after heating. Such ingredients are there, then you need to go for other types of sterilization. So sterilization generally it is done by autoclaving. Okay, this is to avoid microbial contamination. I already told you this medium is somewhat appropriate for microbial growth. So if you place a tissue over this medium, that will attract huge amount of microorganisms, and that organism can grow in a huge rate compared to the plant cells growth. To avoid those microorganisms, we take explant and they surface sterilize it. Also, the medium onto which this explant is kept is also sterilized, and thus that gives a proper combination or condition for the growth of plant cells. We have to maintain a pH for plants growth. Generally, the pH is 5.7. General pH for plant cells. In this pH, the hormones act in its proper way. So you have to maintain the pH by adding the acid and base and maintain the pH 5.7. So these are major requirements of medium. Apart from medium, as I told you, when you keep this medium along with inoculum inside the growth chamber, other substances like humidity, carbon dioxide level, oxygen level, and light are also to be taken care of while growing plant tissues inside the tissue culture room.